All right, so here I wanted to talk about the second part. So, Biochem 1, Chapter 1, Part 2. It is clearly a different day. I'm wearing a different shirt, thus proving that I did not do these back to back. This one should be a little bit quicker because we talked a lot about some of the things that are on this section of the notes, but I did want to go ahead and reiterate a couple of important things. So first of all, we said that living organisms are made up largely of organic molecules. So when we say organic, we mean carbon-based. So especially ask Dr. Eckstead about organic and carbon-based, and carbon makes four bonds and all that fun stuff. So living organisms, yes, they're made up of organic molecules. They're also made up of inorganic molecules as well. So for example, water, H2O, it's not carbon-based, so it's therefore inorganic, and that makes up, anywhere from 50 to 95% of cells. Our cells, it's like a crazy percentage. So most of us is water, which is inorganic. There are also some inorganic ions like potassium, sodium, magnesium, calcium. They're important, especially when you think about them crossing membranes and causing action potentials for our heart to beat and all that. So that's the inorganic component of living organisms. The organic molecules, they're carbon-based. They also usually contain hydrogen, oxygen, nitrogen, and then to a little bit of a lesser extent, phosphorus, sulfur, and then tiny trace amounts of various other elements. So a lot of different elements are in our body, and we can kind of group them into organic and non-organic, but we are considered, since we're alive, to be carbon-based organisms. So the simplest organic molecules are called hydrocarbons. Okay? And so true to their name, hydrocarbons are made up of just hydrogen and carbon. Okay? These, when they're just made up of hydrogen and carbon, and we won't get into the specifics of why, because they share electrons evenly, these molecules tend to be very nonpolar, so, no charges on them. They share electrons evenly, so they don't have dipoles or anything like that. So they tend to be very nonpolar. So if something is very nonpolar, it does not like to interact with things that are polar. Water, H2O, is a very, very polar molecule. Hydrogen and oxygen do not share electrons very equally at all. Also, the shape of water, so you have an oxygen, you have the two hydrogens off of it. You have a lot of electron density going that way and not much going this way, and it's not nice and evened out where it would be canceling each other out. So it means water is a very polar molecule. So non-polar things and polar things don't tend to interact very well. So hydrocarbons, hydrocarbons, they're very non-polar, so they do not interact well with water, which is polar. So we said that hydrocarbons are hydrophobic. Okay. Other molecules that do have charges or do have dipoles or don't share electrons evenly, and they have regions where they're more positive and more negative, those are called polar molecules, which I'm sure we've heard at this point. Those tend to interact very well with water, so those tend to be more hydrophilic. Okay, so hydrophobic don't interact very well with water. Hydrophilic interact well with water. Okay, and this goes a long way into barriers being built among cells and compartmentalizing things and what can cross over and what can't. So the hydrocarbons, just carbon and hydrogen. Once we start adding things to hydrocarbons is where they get a little more complex and more complicated. Okay, so big thing that we're going to have to memorize just right off the bat are functional groups. Okay, you may have seen these already, you may not. Basically, functional groups are groups of atoms that we know how that group of atom behaves, or we know how that group of atoms behaves. So if that group of atoms is attached to the hydrocarbon here, and a certain one is attached here, and a certain one's attached here, we can kind of know, okay, this region's going to behave this way, this region's going to behave this way, this region's going to behave this way. And so we can kind of tell what the overall molecule is going to act like. Okay, so there are a lot of different functional groups out there. There are, let's see, 1, 2, 3, 4, 5 major ones that I want us to know kind of right off the bat. We may add some more, like a thiol, which involves sulfur. But for now, there's 5 that I want us to actually memorize. Okay, and if you go into Canvas, you will see another document that shows the functional groups. Now, what I like to do when I'm in an actual classroom is draw the functional groups up on the board, because I like to write up on the board while I talk. I can't do that, and I don't have, like, I do have a board that my daughter writes on, but I can't hold it up very well. So I just wrote it out on a piece of paper. It's the most, like, low rent, terrible thing that you've ever seen. But it is the functional groups that I want you to memorize. And they are alcohols, aldehydes, ketones, amines, and carboxylic acids. Okay. So if you're looking at the paper where I've drawn them out, so like an alcohol, I have a line and then an OH. Okay. That line, when there's nothing connected to it on the other side, it's assumed that that's a carbon over there, because we're attaching all these to those hydrocarbon kind of backbones. All right. Things like an aldehyde. Okay, we have a carbon double bound to an oxygen up here, and then we have a blank space, and then over on this side we have a hydrogen. Okay, that blank space is assumed that it's bound to another carbon. Okay, so that's kind of important. Just some notation things. So like a ketone, you may see it written as a carbon double bound to an oxygen with 2 carbons on either side, or just 2 lines on either side. It's assumed that those are carbons on there. It makes more sense when you see it written out, if you don't know it already. Okay, so especially when we're talking about carbohydrates, aldehydes versus ketones come into effect. When we're talking about proteins and amino acids, definitely we'll know a lot about amines and carboxylic acids. Okay, and then alcohols they're just an OH group everywhere. Also, just for future reference, don't just memorize, okay, an alcohol looks like, it has a line and then an O and H. Okay, it might be up and down with an oxygen bound to hydrogen. It might be, sideways. So just kind of be able to recognize those in any configuration that they come up on a macromolecule. Okay. Speaking of macromolecules and our biomolecules, we mentioned that there were 4 that I really wanted us to talk about. So we'll briefly go over them again. So we said the 4 are proteins, carbohydrates, fats, and nucleic acids. Okay. So the first one, number 1, proteins. Okay. I go back and forth on which one's the most important, but proteins are certainly the most diverse. Okay. So proteins are made up of building blocks called amino acids. So the polymer is the protein, the monomer that builds them up are amino acids. Okay. Now there are about 20 different amino acids, and they combine in countless numbers of ways to make up, our zillion different proteins. Okay. And remember that a protein form equals function, so how those amino acids come together to form the protein makes it become shaped a certain way, which makes it have a certain job in our bodies. Okay. So there are 20 different amino acids. Each amino acid kind of has a generalized structure. I should have wrote this on the functional group page, but we'll talk more about it as we go on. But each amino acid, they have a general structure. They have a central carbon atom, and that central carbon atom is bound to 4 things, which, again, organic chemistry, carbon makes 4 bonds, always, forever. Okay? But the central carbon atom of an amino acid is bound to 4 particular things. First, it's bound to a hydrogen. Okay? Boring. It's bound to an amine group, which is a nitrogen-based group. It's bound to a carboxylic acid group, which is a carbon double bound to an oxygen and bound to an OH. So you might see it written as COOH. And then the 4th thing that is bound to, just a picture of what I'm drawing in the air here, is called the R group. And the R group for an amino acid kind of defines what that amino acid is. So, for example, alanine, that the R group is always a hydrogen. Okay, if I think it's glycine, I think is the one with the hydrogen. If it's alanine, then the R group is another carbon. Different R groups make up the different amino acids. That's what makes them different. Okay, the R groups kind of determine how the amino acids behave. So we can kind of group the amino acids based on what their R group makes them do. Okay? So we can kind of group them into, these amino acids tend to be polar because their R group is polar. These amino acids tend to be more nonpolar because the R group is nonpolar, even though they have polar regions, but it's more nonpolar because it's got a nonpolar R group. Okay, or these amino acids become more acidic, so they lose hydrogens more easily. These amino acids are more basic; they gain hydrogen ions more easily because of what their R groups do. So, different ways to kind of sort the different amino acids, but there's 20 of them. They come in all different combinations to form our unlimited number of proteins. Okay, when they do join together, the amine of one amino acid joins with the carboxylic acid of the next amine to form what is called a peptide bond. And again, we'll talk more about this when we really get into the protein chapter. Okay. Because of that peptide bond, if we have a long chain of amino acids joined together, we call that a polypeptide. So you may see polypeptide and protein kind of used interchangeably. I think technically that's not true. There's a little bit differences in the terms, but I'm not going to count it off if you call a protein a polypeptide. Okay. So the second macromolecule that we're talking about are carbohydrates, also known as polysaccharides. Okay, so whereas proteins, the macromolecule made up of the monomers of amino acids. Okay, polysaccharides or carbohydrates are the macromolecule, and they're made up of monosaccharides. So polysaccharides, also known as carbohydrates, monosaccharides, also known as simple sugars. So anything you see sugar or glyco something, it's indicating carbohydrates. So the different simple sugars are things like glucose, fructose. We'll talk about what makes an aldose and a ketose, or what makes hexose and a pentose. Different depends on the number of carbons and how those carbons are rearranged. But there's different ways to look at the different monosaccharides. Okay. In general, carbohydrates are used for energy. So we said proteins kind of do everything. Have a wide range of uses and jobs. Carbohydrates, they are used for energy, and they might be used as markers of something. So carbohydrates can join onto the other biomolecules to form different things. So a carbohydrate plus a protein would be a glycoprotein. A carbohydrate added to a lipid would be a glycolipid, and so on. So if you see glyco, think sugar, think carbohydrate. Okay. The third macromolecule is fats, which more commonly known as lipids in the textbook realm. They don't have any particular structure that we can group them all in. So fats include things like fatty acids, sphingolipids, just lipids in general. Okay. For the most part, fats tend to be extremely hydrophobic, meaning they're extremely nonpolar. Okay. So the membranes formed in cells tend to be formed of phospholipids, so a type of lipid, because they do not interact very well with water, so they form a nice barrier that water cannot interact with or cross. Okay. Just for now, just know that there are two different flavors of fatty acids, which are a type of lipid, saturated and unsaturated. So saturated fatty acids do not have any carbon-carbon double bonds. They only have carbon-carbon single bonds. So they are saturated. They have as many hydrogens as those carbons can handle. Whereas an unsaturated fatty acid may have a double bond, so it technically could handle more hydrogen, so we call it unsaturated. Why does that matter? For our purposes, not very much. Just know that saturated fatty acids tend to be, since they only have single bonds that tend to be nice and straight and planar, and so they stack very well. And so, like at room temperature, they tend to be solid. Whereas an unsaturated fatty acid, that double bond kind of bends the fatty acid. They don't stack very well because they're all bent and crooked, and then once it gets to room temperature, they tend to be liquidy. So those tend to be the more oils and things. Okay. And then our fourth macromolecule, the nucleic acids. These are DNA and RNA. Okay. So the building blocks of the nucleic acids are nucleotides. Right? Nucleotides themselves are made up of three components. So this is super important to know. The three components of a nucleotide are a sugar, a phosphate, and a nitrogenous base. Okay, so I'll say it again. The three components of a nucleotide are a sugar, a phosphate, and a nitrogenous base. Okay, so the sugar can be either ribose or deoxyribose. So for DNA, that sugar is deoxyribose. For RNA, that sugar is ribose. So DNA is actually called deoxyribonucleic acid, and RNA is ribonucleic acid. So DNA has deoxyribose, RNA has ribose. And again, once we get more into the in-depth chapters, we can talk ad nauseum about this. Okay. The second component of a nucleotide is a phosphate. Okay, phosphate's phosphate, no particular branding on that. And then lastly, the third component of a nucleotide is the nitrogenous base. Nitrogenous, nitrogen-based base. These come in kind of two groups, the purines or the pyrimidines. So the bases can be purines, which are adenine and guanine. The pyrimidines are thymine, cytosine, and uracil. I always forget which ones belong to which group because it is much more important to know when we're looking at the structure of DNA that adenine always bonds with thymine, cytosine always bonds with guanine. One purine with one pyrimidine and vice versa. And I forget which ones belong to which camp. But again, we'll talk the structure of DNA and RNA at length later. Okay. We said that when amino acids join to form long polypeptide chains, those bonds are peptide bonds. Okay. When nucleotides join to form long chains, then the bonds that they form are called phosphodiester bonds, and we'll talk more about why that is. So side note, DNA, we have two strands formed by phosphodiester bonds, and then the nitrogenous bases kind of cross over, and they're held together with hydrogen bonds. So I just thought that was kind of interesting, and we'll draw lots of diagrams about that later. All right. So there are a couple of miscellaneous type things in kind of the later parts of chapter 1 of the book. So I just thought I would kind of see if there's anything important in there. So I thought, well, we might need to know a little bit about this just for some background information. So we said, yes, there are countless chemical reactions occurring in our bodies, hence biochemistry. Those are aided by enzymes, which are proteins. But the reactions themselves, there's uncountable number of reactions, but they tend to fall into like some very distinct categories. And if you understand, one, then you can understand all the reactions that are kind of that category. So the categories, just kind of broadly speaking, are nucleophilic substitutions, eliminations, additions, isomerizations, and then my favorite, the redox reactions. Okay, so nucleophilic substitutions involve a atom or molecule called a nucleophile and an atom or molecule called an electrophile. So the nucleophile, it has a lot of electrons. It's very negatively charged. It's very anionic. Okay? And it's wanting to give those electrons away. And then the electrophile is something that has, not very many electrons. It's very positively charged. It's very cationic, and it's looking for electrons. So those interact. And basically, when the nucleophile interacts with the electrophile, it gives those electrons and it forms a new bond between them. The only problem is when that bond is formed, another bond is broken. Okay, so if you think about, if carbon is the thing that is being introduced to a new bond, carbon only makes 4 bonds ever. Okay, So if it makes a new one, it's got to get rid of one. All right. That allows kind of one thing to kind of join carbon and kick something else out, and hence why it's called a nucleophilic substitution. Okay. Not super important to know the exact mechanism of it, but just know that that's kind of how a lot of things work when carbon is involved. Okay. So the second type of reaction that we just need to be aware of are called eliminations. In an elimination, instead of an atom being replaced by another atom, if atoms are removed and a double bond takes its place. So carbon, it's bound to 1, 2, 3, 4 things. If it loses one of those things, it still has to make 4 bonds, so it'll double bond. So it'll be double bond and then single bond, single bond. So 1, 2, 3, 4. Okay. So eliminations are just when atoms are removed, but instead of new atoms taking their place, just double or triple bonds are being formed instead. Okay. Additions, I don't know why this is even included in the book, but one molecule, another molecule, they combine to form big molecule additions. Isomerizations, these happen a lot in, especially when we talk about glycolysis and the Krebs cycle and all those chains of reactions. In an isomerization, so isomers are molecules that have the same atoms just in a different arrangement. So an isomerization means that the atoms of that molecule just get rearranged. Nothing is added, nothing is taken away. They just kind of get rearranged a little bit. So usually it'll be in like, okay, this step leads to this one, then those are rearranged so it can go to the next step. So an isomerization, the atoms of a molecule are being rearranged. And then lastly, my favorite type of reaction, the redox reactions. So we do need to memorize this if you don't know it already. There are such things called oxidations and reductions. So an oxidation is when there is a loss of electrons. A reduction is when there is a gain of electrons. So if I have, atom, molecule, if it loses electrons, we say that it has been oxidized. Okay? If we have a different atom, it gains electrons, we say that it has been reduced. Okay, so something's oxidized and it loses electrons, its charge tends to go up. Okay, which makes sense. It lost all that negative electron energy. Okay, if something gains electrons, it's gaining more negativity, its charge tends to be reduced. So like from -1 to -2 or to -3. Okay. You can see where those two can kind of go hand in hand. If one thing is oxidized and then those electrons can go to reduce the next thing, okay, that is called an oxidation reduction. All right, they're combined into one reaction. So instead of calling it oxidation reduction all the time, we could call it reduction oxidation. And instead of calling it reduction oxidation all the time, we can just call it redox. So those are redox reactions. We'll talk a lot about them when we talk about the electron transport chain, when we talk about NADH and NAD plus and all that. We get more into the energy kinetics type things. All right, so I believe that is it for Chapter 1 notes. This talk that I just recorded on a completely different day from my previous video is what we would have talked about tomorrow, Friday. So definitely go ahead and give the notes a look. Go ahead and give a look to the quiz. Send me the answers, please. And then we can kind of move on to Chapter 2, which is still a little bit broad, and then we can kind of get more into the in-depth stuff, hopefully, once we get into a classroom. All right, so as always, let me know if you need any help, if you have any questions. Feel free to call, text, email, smoke signal, however you want to get in touch with me. All right, thanks.